Yeah.
Okay, it's 1:30, so I think we can start. Welcome to this workshop from Lieven Clement on msqrob2PTM. I will not say much more. I will hand the word over to you in case you have questions or like technical issues or something. I don't want to interrupt even with questions. Just raise your hands
and Lucas and Charlotte can help you out.
Yes, and I guess you will be using the.
I will use a cloud service that so on the cloud service you can access msqrob2. So if you basically log in onto the cloud service with your password and name that is in the file, you can access the cloud service, and then you can basically launch the msqrob2PTM.
And you put the information on Zulip?
Yeah, and I also have put the information. All the information is on Zulip as well. And then the msqrob2PTM virtual machine should start to load. So maybe, and then I will give a brief introduction, and then we can start to run the vignette. So, and once that you were able to get the system running, if you click on it, you just will open a video in the browser. And for people who want to work with it offline, you can also download the Docker according to your specific settings. You can pull the Docker, or if you have a Mac machine, you can pull it with the special platform thing, and that should work as well. So I would suggest that everybody is trying to do that, and in the meantime, I will just start kind of on what we will do today. Are there people not familiar with proteomics data here? Okay, so then I will briefly touch base on the proteomics example. Our markdown that we will use today is the msqrob2PTM paired phospho-enriched sample. So that's a markdown that I will use today, and that is generating this particular presentation. So I will briefly introduce how proteomics data is generated so that you have any idea. So what we typically do is that we want to study potentially abundant proteins, and we typically will take a sample, we will extract the proteins from it, but we cannot measure them directly on a mass spectrometer. We have to put them or digest them into little pieces, into peptides, and these peptides are then first put through an LC that we have at the same time to launch to put on a mass. Out of the LC, you will basically take a bunch of these to the mass spec, and first we will ionize the peptides so that they can feel the electromagnetic field. And if you then look to the result of the mass spec, you typically have a kind of a spectrum where you have a mass over charge value, and then you have a peak. And every peak typically corresponds to one of these peptides. So that's it. And typically, the higher the peak, the more abundant the peptide is. But the peak height in this MS1 spectrum also largely depend on how well these peptides are ionizing. So we cannot directly compare between different peptide species because they're in a different way through the mass. And another thing that we have to keep in mind is that we have multiple of these peptides that correspond to the same protein. So that's like one of the issues that we have. So we already have quite some variability of these peptide characteristics. And then the thing that we did for this precursor or for this peptide, we don't know what the peptide species is. And then we typically will take that peptide and we will fragment the peptide in a second round of mass spectrometry to have a kind of a fingerprint, and that is then used for identification. So that's the typical. We typically use MS1 for quantification and MS2 for identification. And then one of the issues that also happens is that sometimes these peptides are misidentified, and I basically attribute a wrong peptide to the wrong protein. So we also have like outliers. So what we have to do, outliers, highly variable peptide quantities. And another thing that we also have is misdata. Sometimes if a peptide does not fly well through the mass spec, and if the protein is not really abundant, we cannot the detection limit of the instrument. So we have messy data, missing data, and all of these things. And that's nice to work on as a statistician. The data that we will use is data from phosphoproteomics. So you can have a kind of a modification group that is attached on a protein, and then the conformation of the protein changes. And then it can have another biological function. So one of the things that we post-translational modifications. And in order to do that, we will basically analyze like modified peptides right away. So that's what we will do in the course of today. We will use a slightly different approach. And in the different approach of the more modern acquisitions, instead of selecting one single peak, and put that in the MS2 mode. So we will have an MS2 spectrum that contains a kind of a hybrid thing of multiple peptides that are ionized, that are basically fragmented together. So the key thing in this more notches is that we will have to do a kind of a deconvolution of this MS2 spectrum to know which precursors that there are in the sample are now able to do the quantum also in the MS2 stage. So, but apart from not focus on the identification and the quantification analysis. And there is like a couple of search engines that give you that output, that do this deconvolution for you. So Spectronaut and Diana are the two most famous or most commonly used softwares that are basically doing this deconvolution, and that are then also giving you a kind of intensity value for every in your sample. So that's what we have, and that's what we will use as the input data. We will use a kind of a data matrix with precursors or peptide species in the rows and with the different samples in the columns. So that's what we will start off. Questions related to that? One of the things that often happens in proteomics, and that we will also do here, is that we are often interested in differential abundance at the protein level, but we do not measure the proteins right away. We measure the peptides, and then we have to do some kind of a summarization from peptide to protein level to make sense of it. In our case, we are interested in this modified peptides, so peptides that have, for example, a phospho group attached on it, and we can have multiple peptide species that have a phospho that is mapping at the same location in the protein. Tutorial aggregate all these precursors that carry the same modification that corresponds to the same position in the protein. We will aggregate them together to get a kind of a quantity for a single PTM at a certain location in the protein. So this is a kind of an overview of what we will have to do. We will first get the data into R. We will do QC. We will pre-process the data. We will normalize the data, and then we first will do an analysis at the precursor level, where we just immediately look at differentially abundant peptide species, and then in a second phase we will also aggregate all the peptides that map to the same position in the protein and have a modification there. We will aggregate them together, and then we will also do a differential abundance analysis at the PTM level. So this is what we will do. And another thing that is also important if you PTMs is that sometimes you find a differential PTM, so a PTM that goes up and down, or that is differently abundant in two conditions, but the difference to the PTM biological function. Sometimes it's just because the protein goes down. So another thing in the analysis that we will have to do is that we'll have to correct for the overall protein about like what we would call differentially used PTMs, and that's what we will cover today. Was everybody able to launch the system? So what I would do now is that I suggest that we first, before we continue, that we first already load the libraries. So if you go down a bit in the vignette, you will see the section where we load where we can load the libraries. So I suggest that we first already load the libraries, and that we also already load the precursor table, so that we already get the data into the software. So the output of this, but there's a delay on the screen. The output that we have from the machine file format is like already compressed files that contain a long list measured by the machine, and then you have information on the intensity for that precursor. You have information on the precursor, so we have a lot of information, like compressed flat tables that we will read in. We typically file cache, so I already have cached the data on the Docker, so that you do not have to download it from the web, so it's already on the computer. So if you just run this, we also have the path ready to the data, and then we also can already load the data into the machine, and then we basically will have like here two different tables. Will have a data table with ordinary precursors, and I will have a data table with precursors that are and PTMs. So PTMs are often difficult to pick up, and in the experimental layout, they typically will enrich the samples so that they have that they are focusing on precursors that already have a modification. So the use of is just this convention that's what they use, and that's like very efficient in ways of compress. That seems to be a very efficient way to compress the files. You don't have been like H5H. Probably it could, but that's the two leading platforms now use. Or it is file, so you can export it in the two ways platforms, but most people nowadays use this Parquet files. Yes. Yeah, this here in the first chunk I was set. Either I was caching the data web on the machine and established a link, and in the second phase with the arrow package you can read the Parquet file, and that is reading the Parquet file in the data. Parquet file, or can you kind of lexical file connected to the file? Like load that as you request, like you have. Yeah, you can probably. A few features like delayed. I don't think you can use delayed information, so we have to get it in the data, we have to get it in the system anyway for the downstream analysis. But that would be a cool thing that we could have in the future, so to work with like single cell experiments, because then the memory usage would be much better. So what is the experimental data about that we will work? So the data that we will work with is data from mice. So we have 6 mice per. We have 4 different strains of mice, wild type mice, and then 3 types of mice where they basically have a kind of a modified RNA messenger RNA molecule that has a kind of a mutation in the in its body, and because of that mutation, it has a proline codon, but it is likely to integrate an alanine instead. So it is basically, and they had like 3 different variants, 2 proline codon RNAs, and one that is building an alanine instead, and the other one is likely to build in serine instead. And then they also have a 3rd mouse strain where they basically had a mutation in the transport RNA that is actually having an arginine codon, but is incorporating a serine codon. And we see that happens about 6 to 7% of the cases. We'll have a number of proteins that are built from mutated strains. And we now also want to see if that has an impact on the modifications that are on the samples. So they have in their experiment, they have 4 different strains, and for each strain they have 6 biological repeats per strain, and then for every repeat they will basically just do a conventional mass spec analysis, and they will do a mass spec analysis on after an enrichment to have more phosphors. And these are the 2 parquet files that we have. We have a parquet file for the ordinary proteomics data. We have a parquet file for the enriched samples. And this ordinary data and the phospho-enriched data is basically all gathered from the same sample. So we have a kind of a paired design. Other questions related to the experiment? Clear what we will with? So now I think that we can to the vignette. There's some lag on. Okay, so we have the data now in the computer, and I can just have a look. I have issues with the Zoom thing here. Okay. So if I just look at the head, so the first. Yeah, it's a typo. I'm sorry about that. I'm a little bit dyslexic, you see. And what do we see? We basically see that we have a very big table, and every row contains a certain feature that has been measured. Here you see the ID of the feature, you see the sequence of a feature, and the modified sequence basically includes the sequence, the amino acid sequence, and it also includes modifications. And here you see that you have a modification of type 1, and the modification of type 1 is an acetylation. And we are mainly interested in the modifications of type 21, which are phosphorylations. So in these sequences, if you have a certain amino acid that has been modified, we will see that as Unimod 21. So that is indicated. Yeah. And then we also have the charge of the precursor. So we have a lot of information. We have information on the run. We have information on the protein ID. So they map the peptides to the protein. So we have information for every precursor from which protein it comes. So we also have the information on the retention time. So we have a lot of information in these files. Because we have so much data, and if you read it in later on in the Q features data containers, all the data that is in here for one line will basically end up in the raw data. So these are informations on the feature that you have measured. And because we will reduce the memory footprint a bit, I basically have some code here where we'll take the precursor table that we just have read into memory, and then I will basically filter one set of samples out. So I will not work with the sample that for which we have a protein that is in the codon and that is incorporated as a serine. I will not use that strain because this strain gave so many differentially abundant proteins that it was difficult to see all the rest if we had that in the vignette. So we will basically work with 3 strains, the control, and 2 modified strains, and we drop one of the strains here. And then we will basically select the variables that we will use. So we will select the run so that we know from in which run this particular precursor has been acquired. We will also get information on the precursor ID, of course, on the modified sequence so that we know how the sequence looks like with the modifications. On the stripped sequence, that's a sequence without the modification. So if you get all the modifications out, you have the peptide sequence, and we can then use that peptide sequence to map it to the protein to see where it comes from in the protein. We also will keep the precursor charge, the protein group, so the group of proteins that for which this sequence is mapping to, the name of the protein, the ID of the protein, and the protein site is also something that is very useful. The protein site is basically including the number of the amino acid, which amino acid position the modification is in the protein. So we basically already have a kind of an absolute, the software already gives you a kind of an absolute position in the protein where the modification is belonging to. And that's very useful for making plots later on, and also for matching different precursors that have a certain modification at the same position in the protein. So this is for us a very important variable to use. And then we have the gene precursor quantity, which is the main thing that we will use for our quantification. So this is a kind of an intensity that you have for this particular precursor based on this MS2 spectrum. They also already have a kind of a precursor normalized. So Diane and Spectronaut have their own internal normalization, and you can already get normalized precursor intensities out. They typically behave quite well, but sometimes it doesn't work well. And I don't know what they do under the hood, so I prefer to work with the precursor quantities and to do the normalization myself. If I still want to use the normalization of Diane, I basically apply their normalization factor. So they basically have a different normalization factor for every precursor, and we can use that for normalization if you don't want to use our own normalization procedures. Then we also have the MS1 area, and that's quantification at the MS1 stage. So if you just have the first MS1 stage, the peak height at the MS1 stage, that is the MS area, and then you also have a normalization there. But typically, we see that quantification at the MS2 stage works better than at the MS1 stage for this softwares. And then they also already contain, already have a kind of a summarized variable. So the protein group max LFQ value is basically the intensity that you have if you would aggregate all the sequences that belong to the same protein. So this is what you would get if you would read in a protein table, so to speak. So this summarization from peptide to protein is there already present. And then we have Q values that are basically Q values that give the quality on the identification of the precursor. And we will also use the LibQ value, because sometimes an sequence has not been identified in this run, but it can be recovered by matching it to the IDs that you have in the other runs. So if you do a kind of a matching between runs, so if you find an identification somewhere, and then in the other runs, we will go and look at the same position. Do we have some intensities there? We can have some matching between runs. And then they also have a Q value for these matches between us. And we will use that mainly for filtering. So we will typically use a kind of an FDR level of identification at 1%. And then we also have proteotypic variable that is basically indicating if this precursor can be mapped uniquely to a protein, because sometimes you have precursors that can originate from multiple proteins, and then you don't really know to what protein it belongs. And we will typically only use a proteotypic precursors in our downstream analysis. And then we have information on the decoys, and decoys are typically used in the identifications. These are sequences that do not exist, but that you use to estimate the Q values. And sometimes some of these decoys are reported, and that's of course not of our interest because these sequences do not exist. And the last thing that we also can use is the retention time. So this is what we do. So we will filter the data first, and then maybe we can already run that chunk. So we will filter the data first, And we will do that for both the precursor assay and for the precursor PTM table. So we will basically keep the two values or the two sets of data. So that's what we will work with. And to give you an idea on some of these important variables that we will use downstream, this protein site, modified sequences and strip sequences, I will just give an overview of this, of the first 20 variables. And what do we see? And it's more clear if I go to the output in the vignette. We basically see here that the protein sites are indicating the name of the protein, and they also indicate the position in the protein. So the second amino acid in the protein is adenine, and then S, I forgot what that is, but it's a serine at the fourth position. So we basically see, and they contain modifications. So the first one contains an acetylation, and the fourth one contains a kind of an phospho, and that's an interesting one for us. And then you also see the strip sequence, so we basically see if you get rid of all the modifications, that's the amino acid sequence of that particular precursor. And that's the data that we will work with. And then here we also see, for instance, a protein that has multiple, this one had like multiple phosphorylations in the same sequence. So it can happen that we have a precursor that has, or that the protein is modified at multiple positions with phosphors, and that's something that we have to work. Are there still questions related to that? So this is the data that we will work with, and we are now interested in finding precursors that are differentially abundant between the different strains, to prioritize modified phospho precursors that might be interesting, might be affected by these mutations. I use a lot of the garbage collection because I have a lot, the data is large. So if we remove data, we will typically try to do some garbage collection. So from the precursor PTM table, I will only, for the precursor PTM table, I will only use precursors that are modified with phosphorylations. I'm not interested in the other modifications. So I will also filter out all the precursors that are not modified. So I only keep the ones that have like a modification with 21 inside. So we can go back to the vignette now. So that this is what we do here. So I subset this phosphorylations. I will just do some garbage collection because the data has been reduced. And then I will also make a kind of a first plot of all the intensities that are in my precursor, that of all the precursor quantity intensities that are the ones that we will use for quantification in this files, just to see what the overall distribution is for the precursors. So these are the regular, this is from the samples that are not enriched. And if we do that, we basically see that we have like abundances here for the enriched, non-enriched samples for the normal proteomics assays and the ones that are phospho-enriched. And the reason why I do that is because the software often already does imputation if the user is asking for that, and then we can see that here in a little peak, and we typically do not like to work with the imputed values, and then we can basically filter them. So this is just a kind of a fast quality check to see have they imputed the data or not. Because they typically impute the data under the assumption of missingness by low abundance, and that sometimes gives us wrong results if you use that downstream. Yes?
Are these two x-axis is it true that the PTM ones tend to be lower density?
I can imagine. So PTMs, it's more difficult to pick up PTM. So I can imagine that like the overall abundance of the proteins that we of the features that we have or that we pick up in that assay is indeed lower. So it can all, yeah. So I have not thought about that. So, but yeah, I could imagine that that could be one of the reasons. now that we have the data in the computer, we also need to have like the sample annotation ready so that we know which samples we have in this long table. And we can basically extract that annotation from the run labels. So the researchers did not provide a nice annotation table, but we could luckily extract it from the sample names. And here I will just take the precursor table. I will get all the distinct run names out of it, and then I can split the run names according to the separate the run names according to the separator underscore, and that basically gives us the name of the run, a kind of a thing that I don't know what it means, so I call it label 2, and then the strain, the repeat for the strain 1 up to 6. The type, is it like a normal protein, a protein type of the phospho-enriched thing, and then also the way of acquisition, because they both had data with data-independent acquisition and data-dependent acquisition, but for the vignette, we only use the data acquisition. And then what we also have, if we use an annotation file and if you have a long table, we will later on use qfeatures. And if you use the read qfeatures function, our annotation file should contain a run column, and that run column is then basically indicating this is the name of the run that you can find back in this big long table to aggregate all the features together, to collapse all the features of the same run. And then I also add a kind of a sample ID. The sample ID is like a unique identifier of the samples. I could have used the run for that, but that's a very long name. So I will basically just paste the strain and the repeat together. And then we have a short sample name that I will just use later on for plotting. And then I will arrange the values according to the sample ID so that the sample IDs are nicely in alphabetic order. And then my columns will also have been in nice alphabetic order if I read and stuff later. And I will do the same, so I will just do that here, and then you will see the results. So we basically see that we have 18 different samples. So we have 3 strains times 6 repeats. We have information on the run, on the label, on the strain, on the repeat, on the type, and the acquisition. And then we also have here the sample type that is indicating the strain name and the repeat. And we can now do the same thing for the phospho data, and then we basically see that here we have the phospho-enriched samples, and then we have the same information. And actually, these come from the same biological inputs. The phospho and the non-phosphos, the phospho-enriched come from the same samples. Is that clear? And now we have everything set up. We have our initial precursor table. We have our annotation data, and we can now convert that long table in a kind of a qfeatures object. That's an object that has a nice data structure to work with in proteomics. So this is what we will do next. So I will use the read qfeatures function in order to read in the data. And I will tell you more about the data containers while we are actually converting the long table into a kind of a qfeatures object. So I suggest that we just hit the run button, and then I will, in the meantime, go back to the presentation to tell you more about the data structure. So the qfeatures data structure, there's also a very nice poster to it. Oh, I'm a little bit too far already. So what it is using. So it is basically a kind of an extension of the multi-assay experiments class, and it can contain multiple assays. And we will typically read in precursor data here. So we have our precursors in the rows, we have the samples in the columns, and then if we summarize, if we filter the data, or if we normalize the data, each time that we do a manipulation, it basically will make a new copy of the assay so that we can keep track of the raw data up to all the manipulation steps that we have done. So if you find something that is differentially abundant, we can then go back to all the different intermediate steps to see if this is supporting what we find later on. Another thing that is nice, if you take the precursor level data and if you would summarize it into proteins, for instance, then there are links between the assays. So if I pull out a protein out of the Q features object, I can immediately also get all the precursors that gave rise to this protein level value. That's very nice. We also have row data with information on the features and column data with information on the columns. So with msqrob2, the software that we have, the results of the inference will also be stored in the row data. So we will then have like one object that contains all the data manipulation steps with the intermediate data and then also the final inference together in one big object. And that's very nice for reproducible. So we can now go back to this studio server, and what have we seen? We basically have seen that we will read in two different files. I will read in the precursor data, so I will have one Q features object that I have read in for the precursors. I have a second Q features object that I read in for the PTM's precursors. I use the annotation and the annotation PTM as the column data. I also use the precursor quantity as the quantification value that I will use. And I will match the run column in my call data to the run variable in the precursor table that I use to read the data in. And I will use the precursor ID as my feature name for my feature. So that's what is done. So I have two different Q features objects. They are in a list. And then I will reduce the list with the C function. And if you do C function on Q features objects, you can just merge them together in one big Q features assay. So I will basically now have a Q features object that contains 18 plus 18 assays. So because I have a long table, every run is stored in a different assay. So that's what I have now. So I have 18 normal proteome assays, and I have 18 phosphor assays. So that's the data that we will work. I will now remove the precursor and the precursor PTM table so that I will free some memory, and then we can continue. In the first steps, before we do the data modeling, we will first have to do pre-processing. And I will first replace zeros that are missing values that were not picked up by the machine by NAs, because if I log transform the data later on, I don't want to have a log of minus infinity, because we typically transform the intensities because they have a kind of a mean variance relationship, and the variance is stabilized if you log transform. Then we will filter the precursors, we will do log transformation, normalization, and we will also do summarization for the non-enriched assays so that we have protein level summaries for every protein in the experiment. And this is something that we will only do for the non-enriched runs. For the phospho-enriched runs, we will just work with precursor data right. So we will use the Q features functions for that. QF is the Q features object that we have stored. I can use the 0 as an A function to replace the assay values that are zeros into NAs, and I can do that for all the different assays that are stored in the Q features objects by simply specifying all the names of the assays that are there. So if you do that, we basically have now all zero NAs where we had zero values. If you want to, you could impute the zeros, but that's something that we typically advise against. But if you really would like to do that, you can use the impute functions from the Q features package. They just provide them. I will not stop you from doing that, but it's typically not a good idea. The first thing that we will do is that we will remove precursors now that do not make sense. So some of the precursors are questionable because they were not identified with a very good FDR, or they are mapping to like proteins that have multiple, or they are mapping to multiple proteins, or they are belonging to these decoys. So we will first do a kind of a standard filtering, and we will typically filter. We will only keep identifications at a 1% FDR. We will remove precursors that were not able to be mapped to a protein. Then they have an empty precursor ID. They have an empty string. We will also filter out the decoys, and we will only keep peptides that match to a single protein. And that's what we do here. So we can use the filter features function for that. So we take the QF object, we pipe it to the filter features function, and we will then apply a filtering. We will take the Q value, we will insist that the Q value is below 0.01. Also for the protein group, so we also have a Q value at the protein level. This also has to be 0.1. And also for the ones that are matched between them. And these are the default values that are also advised by Diane, and we take that over. We also remove precursors that were not matched to any sequence, and we remove we only keep features that do not match to a decoy, and we also only keep features that are proteotypic. And by having this AND statement, we basically have a long true or false conditional thing that we generate. And if you do that, we will basically filter out a number of things. So now the filtering has been done, and we now have like good quality precursors to work on further. Yeah. Proteotypic. A proteotypic means that a proteotypic precursor is a precursor that is only mapping to a single protein. So you do not have an ambiguous. You do not. Proteins are precursors that map uniquely to a protein or proteotype. Yeah. Yeah, that's related to a specific species. So what you typically do is that you have your mass spec data, and then you also download from UniProt for your species. You download the whole FASTA file. We will do that also because we will need that later on to make nice plots for proteins. So you basically have a kind of a reference protein sequence of all the possible proteins for this particular species, and you use that for the map. So that's what it's. Okay. I now forgot. Yeah, I have invoked the command. So we can now now we are in the next step. We now have good precursors, and we will now put all the precursors that we have for the normal assay together in like a data table that contains features in the rows and that contains samples in the columns, so that we have like one big data table for the protein for the normal assay, and that we have one big data table for the PTM assays. So we will basically group the individual assays together so that we have like a kind of a data table structure with features in the rows and with samples in the columns. And we will basically join the assays together according to their name. So all the assays that have proteome in the name will be grouped together according to the precursor ID. So if they have the same precursor, they will basically end up in the same row. And I will store them under the name precursors. And then I will also make a second assay where I join all those phospho samples together according to their precursor ID. And I will call that assay precursor PTM. So by doing this, I basically make two new assays, and this takes a while because it's stitching all these things together, and that will allow us to have like a kind of a nice data table structure.
Are we always supposed to do the garbage collection, or just like?
No, I had to do it here because the machine was crashing. So the Docker was crashing because I was running out of memory and I was too lazy to identify where it happened. So I just added garbage collection a lot. But on my own normal machine, I never use the garbage collection. It's just to be sure that I was not having issues here So we basically see now that we have two new assays, a precursor assay, and that now has 88,000 features and 18 different samples for the regular samples, and then we have 59,000 precursors that are phosphorylated, and I have again 18 samples for that as well. And we can now safely remove the other data to reduce the memory footprint. On my local machine, I typically don't do that, but here for the Docker, I also have decided to free some memory to keep the the Q features object a little bit lighter. What? Because I never have trouble with the memory. I would do that for single cell data for sure, but up to now, if I'm just having like typical bulk data sets, yeah, and then I can still retrieve all the information that is in there, but that's what we typically do. And now I will filter out precursors that are not picked up in many samples. So I typically would like to have precursors that are picked up at least 6 samples in order to reliably identify or estimate their features, characteristics. So we will basically keep a precursor if it is picked up in 6 out of 18 samples. And we do that with the filter NA function, and it is basically calculating the proportion of missing values that you allow, and I allow 12 on 18 missing values for a precursor. And then we basically see that we have kicked out about 20,000 phosphopeptides precursors because. Often, if you quantify or if you summarize the data from the precursor level to the protein level, in many cases, collaborators that we work with are only trusting the protein abundances if they are based on multiple precursors. So if you have at least 2 precursors for a protein, they trust it. And then you can filter out precursors proteins that were only matched with 1 precursor as a kind of a safety thing. Here we will not do that because we will use the protein level summaries to correct. We will correct the precursors, phosphoprecursors for the abundance of the protein. And if I get rid of the protein, I cannot calculate the usage. So I will be a little bit more lenient. I'm normally, but I just all the scientists, all the wet lab scientists that I work with in Ghent are often in favor of doing that. So I just provide it here, but it's up to the user to filter it out. If it's reliably identified and there, it's just something that is then kind of a quantity at the precursor value instead of at the protein level value. Yeah, it's up to the user. But another thing is that sometimes if you have a single precursor, it can be a kind of a mismatch, and it's just to be safe with respect. And now we can log transform the data, so we can use the log transform function. We can again have the assay as an input. We will log transform with base 2. We will do that for the precursor assay, and we will store the result in the precursor underscore log assay. So we will make a new assay with log transformed values, and I will do the same thing for the PTMs. So we now have log transformed data. Yes. I should check. Probably Leopold knows. So now we have log transformed data, and now we can start to look at the distributions of the intensities to see if normalization is required. And this is what I will do here. I will take my Q features object, and this with Q features objects, you can do 3-way subsetting. You can subset on the features, you can subset on the columns, or you can also subset on the assays. And you can also, if you take a C with multiple assays, then you can, for instance, select the protein name, and then all the features that match to this protein name and that are linked are kept together for all the features. So this is a very, very easy way to subset your data. So here I will only take the precursor log assay. I will then convert them in a long format to do plotting. I will convert them in a data. I will also add the column. The variables in the call data to there, so that I can color them according to the sample. I will filter out all the values, the assay values that are NAs, because I don't want to have them anyway. And then I will do a ggplot, and I will plot the value, and I will color them according to the strain, and I will group them according to the call. And then I will have and I will then plot a density for every sample. That's what we do here. These are the results. And if you look to the results, and I do the same thing for the PTMs, we basically see that the data is already very, very clean. So I see that we will need normalization, but the data is already very, very clean for being not normalized, both for the regular non-enriched data as for the enriched data. So that's what we basically see. And now we will normalize the data. We typically do that by shifting the curves so that they nicely align. So we have different ways to do that. A common approach is median summarization and normalization. But what we have seen lately is that using a kind of the DSEC2 method for the normalization works very good in proteomics. And what is DSEC2 doing? It is first calculating a reference sample by taking the geometric mean of all the features. And then it is calculating log-fold changes. And in the meantime, while I'm explaining it, I will just do the normalization. It is calculating log-fold changes for every sample with respect to the reference sample. And it is then taking the median value of this log-fold changes as a kind of a normalization factor of the log scale. And that's what we get. So with the NF log median of ratios, you basically get like this log-fold change, the median log-fold change for every sample as compared to the reference sample out of it. And we can then sweep them out of the assay. So we can basically sweep over all the columns, and we can each time subtract the median of ratios from it to have the normalization. And we do that both for the Q features, for both Q features objects. And then we can I probably already have done that, right? And then we can look to the results of the normalization. And we basically now see that indeed the curves are nicely aligned for the normalization, after normalization. So we now have nicely normalized data. What I also typically do as a kind of a QC thing is that I typically also look at the normalization for all the features that all the samples have in common, because we have missingness that is different between the samples. So I also look at the common features to see if it also makes sense for the common features. And here we clearly see that also for the common features, we have nicely normalized data. And this is not always the case. So sometimes if you do median normalization, you will see that it does not work well for the common features if there are large differences in the missingness. Yeah. So what I basically do, I take my assay values, and you can see what I have done here. I take my assay values and I do NA exclude, so I only keep the rows that are full. So that's what it does. So this is what the code does, and that's like a useful thing to do. And the last step that we have to do is summarization. Yes? I have never worked with spiking controls. I first would look to them. I would look to them. It would be nice to have them. And now we will summarize the data of the regular sample from precursor level values and protein level values, because we would like to use these protein level values to see if the protein goes up and down in a different way than the phospho precursors. And I can do summarization. So I will first invoke the summarization, and then I will then explain what the summarization is doing. What are we doing? What we typically do is that we use models that take both the peptide species into account and the sample effect into account. So we basically will calculate a summary at the protein level while correcting for peptide species. So that's what we do. And if you do that, we can correct for the missingness and for differences in the peptide characteristics. So that's what we normally use. A common way to do the summarization for Diane and Spectronaut is that they often use the max-LFQ value, the max-LFQ approach. And the max-LFQ approach is not really using a kind of a model-based approach. With max-LFQ, you are basically first calculating for every sample, for every pair of samples, you are calculating the log fold change based on their common precursors. And then this is like this log fold change that we calculate for the common precursors between two samples, and we will then estimate a kind of a protein level abundance value that is respecting this differential expression between the common precursors as good as possible. So that's what they do, and this gives more or less the same results as a kind of a model-based approach where you have a sample effect while correcting. And then we basically have finished the whole normalization. And then we see that we have information on 4,607 proteins for this normal samples. Is that clear for everyone? So now that we have finished that, I normally would do the QC, but I will skip the QC. You can look to the QC yourself later on. We already have done a bit of QC by looking at the density plots, but we are now ready to start with the model. So I will first, that's what the researchers are interested in. They are interested in finding or prioritizing differentially abundant precursors that are phosphorylated. And I will first do an analysis where I do a classical differential analysis on this precursor. So I will fit a model row by row for the precursor matrix, and I will calculate differential abundance between the samples. And we typically use msqrob2 for that. And if you use msqrob2, we basically use a model and a linear model. And with a linear model, we have to define the model, and we typically can model multiple sources of variability. Here we have a simple experiment. I only have one source of variability, and that's a strain. I did not have additional covariates at my disposal, so we will basically model the means of the samples with three different sample means, with three different means for every strain. If you have more complex designs with multiple factors, we can just use conventional LMER formula for that. So, and in the poster, you have a complex example with random effects, fixed effects, and so on. Yes? I will do that later on. I don't do that deliberately here because we will sometimes see that some of the precursors do something else than the average PTM. And this is because they can also have other modifications there. But I typically first like to do an analysis at the precursor level, and then we will do an analysis at the PTM level, so that we have it at different resolutions. So you're already poking one way, so you're a little bit too fast. So we take the we will make a model. I only have to specify the right-sided formula. So I specify, I have the tilde for the formula, I use the strain, and then I can use the msqrob function that takes the Q features objects. It will work on the precursor PTM norm assay with the normalized values, and it will fit a model to every row. And we also use robust regression. So in msqrob2, you can also downweight outliers with the M estimation, and that gives you more robust results. And this has been carried out. And now we can do the statistical inference. So we can now start to do hypothesis testing. And we also use the nice package of Charlotte with the Explore design matrix that has made teaching much more easy for me. So we basically see that we can model precursor level data with a model, and we will model three different means. A mean for the arginine serine strain, and that mean is captured with the intercept. A mean for the control samples that's equal to the intercept plus the strain control, and then we have a mean for the proline alanine samples strains, and this is the intercept plus strain proline alanine. And we can now think about differential abundance. If you want to have differential abundance between the control strain and the arginine serine strain, we basically see that this is this mean minus that mean, so the log2 fold change is equal to the strain control model parameter. And in msqrob2, you have to specify the null hypothesis. So I would specify the null hypothesis that strain control is equal to 0. And that so we will have three null hypotheses. Strain control is equal to 0. Strain proline alanine is equal to 0 for this contrast, and then I still have the difference between the strain proline alanine and the strain control, and that's like the third fold change. If you only have a simple design, we also have a kind of a thing that is generating this hypothesis with the create pairwise contrasts. You will make all these contrasts automatically, but researchers can also define more complex contrasts. For instance, if you would like to compare this versus the mean of the two others, that's also possible. So you can define the contrast as you find relevant. You see that if you use that, we basically have like these three null hypotheses that are generated automatically. And after you have generated the null hypotheses, we can use these null hypotheses to automatically make contrasts for all the hypotheses that we want to test. And I also have to give in the parameter names of the model, and I can extract them from the design model. And then we basically see that we have a contrast matrix that will that we can use to do statistical methods. And now you can do hypothesis testing with msqrob2 by simply using the hypothesis test function, and that is calculating all these hypotheses for each of the models, and the results are stored in the raw data. The models are also stored in the raw data, so everything is nicely available in the object. And we again do that for the precursor PTM norm assay, and then we now can start to look at the results. So we can basically use the msqrob collect function to collect the output of all the tests that we have done and to store them in one big data table. And then we have a table. And we can use the table to make result tables, and I will use an FDR level of 5% to work with. And we can now, for every contrast, I can take the inference PTMs, and I will filter them if the adjusted p-value. I will filter the features for which the adjusted p-value is smaller than the significance level, and for a certain contrast. And then I will basically plot this, and this gives me three different tables. A table for every contrast. And then I have in this table the log2 fold changes, p-values, and so on. We can also use the results to make plots. So I can also make volcano plots. So if I use a plot volcano function on a results table, I make a volcano plot, and I can also use a facet wrap with the contrast so that I have a volcano plot for every contrast. And then I will also show the results. And then we see that we have a high number of differentially abundant features for every contrast. So we see that we have a lot of differentially abundant phosphorus. In this experiment. I can also count how many differentially abundant phosphols that I have for every contrast. And I basically see that we have, for the first contrast, where I compare the control samples to the strain alanine, proline alanine serine. We basically see that we have a bunch of downregulated and a bunch of upregulated features. And we see that for the last contrast, where we compare the proline alanine samples to the control samples, that we have more than almost 400 differentially abundant phosphorylated precursors. And that seems to be a lot. We can now also make plots, and this is a nice thing of the Q features. I can first make heat maps, but yeah, I think that most of you are familiar in making a heat map, so I will not. So we can have nice heat maps, and we clearly see that we have blocks of upregulated and downregulated features according to the conditions that we have used. And the nice thing of Q features is that we now can also explore the results for individual features. And here I will first look to the most significant feature that we have. I will just identify the most significant feature, the feature with the minimum p-value, and I will pull out the name of the precursor, and then you basically see that we then have log fold changes and standard errors for this particular feature. And then I basically have it for all the three contrasts. So that's what I have. And then I can make plots. So for this particular feature, I will make a plot, and I will extract that feature from the QFeatures object. And I will return both the precursor data that is not normalized and the normalized precursor data. So I will basically be able to make two plots next to each other, one with the data before normalization, one with the data after normalization. We again use this long formula, and we'll make a plot where we will plot the values, the intensity values in function of the sample ID. And I will also add a line plot to it so that I see like how all the features are connected to each other across the groups. And this basically gives me two plots, a kind of a plot where I see all the intensities for all the samples. And we clearly see that it is obvious that we have a highly differentially abundant precursor here, because we have like much higher levels here. So this makes sense, and this was already there as well before normalization, but we see that after normalization that becomes even more obvious. So that's what we have. Now we could say, okay, we can stop here, we are happy, we can just return phosphorylated features, but it's not really sure that they are relevant to our things that we study. It's possible that this thing is differential, not because of the PTM, but because the protein overall goes up. So it now makes sense to also make plots where we add the protein expression values next to it. And that's what I will do here. I will now make 5 different plots for the 5 most differentially phosphoprecursors for the first contrast. And I will add the precursor data, and I will also extract the protein intensity values for the corresponding protein, for it. So I will first extract the top 5 most significant PTMs by taking the inferences, filtering them according to the first contrast, arranging the p-values, and taking the first most significant hits and storing their feature names. And then I will loop over it, and I will make plots, and I will extract only the normalized precursor data from it. And then I will also pull out a protein group, and I will now take the protein assay, and I will then also pull out that particular protein from the protein assay data. So now I have peptide data, precursor data, and the corresponding protein, and I will combine them in one table, and I will make plots. And now I can look basically at, we can make that plot. And now, what do we see if we look to the first feature? Our first feature that seems to be very differentially abundant. We basically see that the feature is doing exactly the same as what the protein is doing. So this feature is not probably differential due to the phosphorylation, but it's likely that it is just going up and down with the protein. So that's not something that is interesting. So we would have to do something else. And is there somebody who has a suggestion? So what could we do? We could basically look at or try to prioritize log fold changes for the precursor after correcting for the log fold change at the protein. And that's what we can do. And then we get in the same thing as what we see if you do differential usage in a transcript level analysis. We can now do differential PTM usage in the context of proteomics. And that's exactly what we can do. But before we will get there, I think it's not really necessary, but it's also interesting to just see how many proteins that are differentially abundant. I think it's just a sensible thing to do. And that's not a lot of work. We can do that analysis by just taking the same steps using the same model, but now just taking the protein level assays out of it, and then we can use the same model for the protein for the proteins. we can then also, after that is fitted, we can again use the same contrasts for the hypothesis test. I do not have to respecify my contrasts; they stay the same. And then I can also collect the results. We can make some. I will not make but we can make some. I can make the volcano plots. So we basically see that also for the non-enriched samples, we see that we have a huge number of proteins that are differentially. And it is clear that it makes sense to correct the precursors for protein level differential abundance so that we have features that are prioritized because of their biology and not because of protein goes. And that's what we will do in the next part of the presentation here. I will now calculate phosphopeptidoform usages. So that's the next thing that we do. Do you still bear with me? Okay. So the key thing is that we will define the usage, the log2 transformed differential usage as the log2 transformed fold change at the PTM level, at the precursor level, minus the log2 fold change at the protein level. So that's what we are after. And then we have usages. And we can calculate the usages in different ways according to the designs that we have. If you have paired samples, the most sensible thing to do if you have a paired design is to first immediately calculate relative abundances at the precursor level. So I take my precursor levels and I subtract the protein intensities of it. If I now would calculate log fold changes between samples, you basically would see that I would have the log fold change of the precursor minus the log fold change of the protein. The nice thing is if we first calculate these abundant usages, that our standard errors that we get also take all sources of variability into account. So if you have a paired design, this is the way to go. And that was surprisingly not clear in the literature yet that was the way to go. MS-Stats, for instance, always calculates differences between the log fold changes, even for a kind of a paired design. And then they lose, of course, some power because they cannot estimate the standard error so often. If you have unpaired samples, and that sometimes happens as well, because if you do not have enough material to do the two runs, if you have unpaired samples, we just suggest to do the precursor level analysis, the protein level analysis, and then calculate the fold changes between the unpaired samples. And then we can just easily, and that's maybe more clear here, I can just go to the usages. So then I can simply calculate the standard errors by just taking the variance of the PTMs plus the variance of the proteins and taking the square root of it because it are independent. And then I can also use like scattered weight approximations to estimate the degrees of freedom. That's a kind of. And if you want to know how this has to be done, I also have a vignette in the for unpaired samples. So I took a subset of 3 samples for phosphos and the 3 other samples for the remaining data. So I have also a vignette on that. Yes?
After no modification whatsoever, and then you produce level you have modification.
Yeah, that's. Yeah, then I would go for differential detection. And in msqrob2, we can use a hurdle model, and that fits 2 components: presence, absence, and differential abundance, given that abundance occurs. And then yeah, it's a separate. Then instead of using the msqrob function, you use the msqrob aggregate or hurdle function. So that's what you do. So we will first calculate the usages, and I have developed a function that can calculate the usages for you. So it will take a summarized experiment of the log2 transformed precursor ions as an input. It takes a summarized experiment for the non-enriched samples into account, and it has an argument for the column with the protein name in it, and it has an argument for the column with the protein name in it for the summarized experiment for the non-enriched samples, and it also has a kind of a variable that is mapping the columns of the precursors to the columns of the protein. So that's what this function needs, and then this function is basically calculating a kind of a normalization factors, or it is getting the log2 transformed abundance for the corresponding protein. And it is first matching the rows of the precursor table to the rows of the non-enriched table at the protein level. And it is then, for all the IDs with a match, it is then actually filling in this kind of a normalization factor according to the protein level thing. And it is matching the columns of the precursor level data to the columns of the protein level data. And now that we have this function, we can carry that out. So we can basically get we will first match the samples. So I will match the sample IDs in the PTM assay with the sample IDs in the protein assay. And then I have like the matched samples. And now I will calculate this normalization factors or these protein values that correspond to the precursor level values by taking my summarized experiment for the PTMs, the summarized experiment for the proteins. I will use the protein group variable in the first assay and the protein group variable in the second assay to match the features to each other. And then I will use these matched samples to match the columns to each other. And then this function will calculate normalization factors. And then I will have a matrix with the same dimensions, the same number of columns, and the same number of rows as my features with the protein level intensities. And I will then sweep them out over the two margins. And that will give me the normalized value. So I will do that, and then I will answer your question. Oh, yeah, I have forgotten to run the function. So I run the function, and then we can do the matching. Yes?
Why did you not normalize it?
So what I do, I have my precursor level abundances, and here I calculate the protein level abundances that correspond to these precursor values. And then if I subtract the two arrays, I have my abundances corrected for the protein abundance. So that's exactly what we do. And it's similar to what you do with normalization. It's a kind of a normalization, but now according to the protein level abundances. And now I will only keep, so I will have some precursors for which I did not have a protein value. That can happen. So I have a little bit, it can happen that I have a bit more missing values now. So I will again filter out precursors from that assay that do not have enough observed values. And now that we have that, before we do the data modeling, we can now again look at the abundances, the densities for the abundances, and we see that they are still very nicely normalized. So I trust them to be comparable, so we can basically go to the next step to model these abundances. Modeling the abundances is very simple. I can use the same code again. I only have to replace my assay with now the assay that contains the usages. So that's the only thing that I have to change. But I can again use the same models as before because I only I have the same mean structure, of course. So I can also use the same code for the inference again. And maybe while it is running, because that takes a while, I can just show you the results of the inference because it's more or less the same way as what we had before. So I can then again report the results with the same code, and we can make volcano plots. And we see that indeed we have much less differentially abundant used features, and we see that they dropped like it's almost 1/3 of what we had originally. So we see that a lot of these precursors were differentially abundant because of the protein, and if we correct for that, we basically have things that are biologically relevant because of the phosphorylation. you also see that if you look to the heat maps, and what we will now also do is that I will also make the plots again, and I will make similar plots as before, but now I can plot both the normalized, I can extract the normalized PSMs together with the usages. So I can see what happened before if you did differential abundance for the top features, and I plot the usages next to it, and I will plot the protein values next to it, so that we basically see what the whole workflow did. So I will extract for a feature the corresponding protein values, and I will then merge them together, and then we can basically make plots again. And we see, for instance, for this peptidoform, we basically see that the normalized values and the usages are very similar because the protein is more or less stable. And if you do that again for the 5 most differentially abundant precursors that were differentially abundant, the same features that we have looked at first, I can now plot the data at the precursor level for the normalized, to say the usages. I extract the proteins of it, and I can also put the p-value for the differential abundance and the p-value for the differential usage in the plot. And then we basically see that for this first feature, if you just look at differential abundance, it's highly significant. But after correcting for protein level intensity, we see that the usage is just more or less random. And then we see that the p-value is bumped up to 0.7. So we basically see that that effectively is doing what it is supposed to do. And then we see that for other features, it doesn't change much because the protein is not differentiable. Questions related to that? Yeah. Yeah. Of course. So what do you expect? That's what I mentioned. It's a super clean sample. And then you have to think about normalization, or you have to go back to the researchers. That's something that is happening, bothering me often with public data. You often see like these pathological cases, but if you consult the technicians, or the researchers that gather the data, they often know why it is, but you cannot get it from the metadata. And then, yeah, if you still continue, you don't know, is it because of normalization that went wrong or whatever? You could try to normalize, but maybe you would overcorrect. So if I see pathological cases, I always like to go back to the technicians. But yes, we see many. And the last few minutes I would like to use for what you mentioned up front. Why do we summarize the PTMs from precursor level to PTM level? And that's what we will do in the last part. In the last part, I will indeed do a differential analysis at the PTM level. So all the precursors that match phospho at the same position in the protein will be collapsed together and will be aggregated together. I will not run the code because it will take us over the break. So what will I do here? I will first make a kind of a new assay, and I will duplicate the rows for features that contain multiple phosphos, because I can have a precursor that contains multiple phosphos, and I will make a new assay that is basically copying the row if it contains multiple phosphos, because then I can aggregate them according to the phospho position of a specific phosphor. So that's what I will do. I will extract the usages from the assays. I will extract the rows, row data from the original values to get rid of all the modeling that I have, all the model columns that I have added. So I don't want to duplicate these. I will now add a new variable with the modification type of the modified features. I will also add a new variable with the location in the original protein. This is with regexes, and I typically have to look. This is one thing where I use ChatGPT or another AA bot for, because I always forget these things. but I explain them here what they are actually doing. So I will basically here for the position, I will get rid of this first part, and then I will basically extract. I will make a string that contains A2, S4, S4, and so on, and then I will split that up so that I have the different columns. And I will also get rid of the first amino acid there so that I have the sites. And then I will replace the row data. I will make duplicates and so on. So this is a kind of a whole procedure to have like an assay that contains multiple rows if I have multiple multiple phosphors. So here I will basically copy the row number as many times as I have phosphorylate, as I have modifications, so that I basically can then use this ID to make a longer matrix, so to speak. And I will also extract the positions by enlisting all these positions. Then I have all the positions for the different assays, and I can now make a kind of a new assay with unnested usage, is what I call. And then I can aggregate the data based on the protein group, and I have made a new variable where I include the protein group, and I add to that the position in the protein, so that I basically can aggregate them according to the modification at that position. And this is what I do here. I just do aggregation as in the same way as I go from precursors to proteins. I now go for precursors that have the same modification to one summary. And then I basically can, again, look at the distributions. They seem to nicely overlap. It's not pathological. I can now do the same thing again. I can do model estimation as before. I can do, I can look at the results tables. I can look at the volcano plots here, and I can also look at how many features that are differential. And we see that the number of features dropped further, but that's because a lot of precursors are now matching to the same position. And the last thing that I also have provided are like a long protein mapping. And I think this is rather cool. We can now represent the result for one protein, and I can indicate all the phosphosides that were detected by precursors at a protein, and I can basically indicate if they were significant. And I can have a kind of a barcode, so to speak, for a protein. And how do we do that? We get the FASTA file that we used for the mapping. Normally, you have to take the FASTA file that was used for the mapping, but the researchers only provided their mapping results without the original FASTA, and I could not grasp what they used. So I used the most recent FASTA, which is suboptimal, but that's what I have to deal with. So I actually download the FASTA file right away from Uniprot. I read the FASTA file in R, and then I basically make a new data frame with the name, with the sequence, and then I also calculate the length of the sequence so that I can make plots. Yes? The reason is that I'm not familiar with biostrings. So if we talk together, help me out to make the vignette better. Yeah, okay. I'm happy to get that feedback. It's the first time that I work with strings. So that was my humble solution to the problem. Anyway, now it is maybe getting even worse. So I will now get the protein. So I basically take my target feature, and the target feature is a protein name with the modification position after it. So I will remove the modification position of it so that I only have the protein name. And I will basically take my PTM usages. I will keep all the PTM usages that map to the same protein, and I will then join them with information of the position. Wait. I will join them. I will join them with the protein modification and the position. So I basically, in the inference table, I will basically add a position, a variable with position to it. And then I will also mutate it. So I will make a new type. If the p-value is missing, I will call it missing. If the p-value is lower than 5, the FDR value is lower than 5%, I will, and if the log4 change is lower than 0, it is downregulated, and otherwise it's upregulated. So I basically have a type. And then, and if it's not none of these cases, I will call it non-single. And now I can basically make a kind of a bar plot where I will plot the position in the position in the protein, and I will color it according to the type. And then I basically get this type of a bar plot for the different contrasts. And we see that here we have downregulation in the control as compared to the alanine serine modified thing. We have some sites that are upregulated in the proline alanine as compared to the alanine serine. And we see that we have the upregulation for some PTMs in the proline alanine instead of the control state. So this is a kind of a summary that the biologists that I work with are liking to see. Yeah, okay. So, yeah. Yeah, okay. So that's it. And the last thing that I have is just the same thing, but for the precursors. But the code is. Thanks for being here and bearing with me. I know it's a long vignette, but I hope it's useful.
Now there's a break and then there's short order.
Yeah. Yeah, sure.